What is interesting about these disorders is their history. If we consider autism, which is a program we did recently, that was discovered 60 years ago. We've had beautiful, detailed, insightful descriptions of Parkinson's disease and Huntington's disease that go back over 150 years. James Parkinson, in 1817, described 6 families that had what he called shaking palsy. The 3 defining features of the disease are that they have a tremor at rest, they have an abnormal posture, and they have muscular weakness. This description was so detailed and so excellent that physicians soon were able to characterize the disease and see exactly what Parkinson saw. And they renamed the disease from shaking palsy to Parkinson's disease. But it was a long time before we began to understand what some of the mechanisms underlying the disease was. It was only around 1960 we began to realize the disease is caused by the fact that there's a death of dopaminergic neurons in the substantia nigra. Dopamine is a modulatory system in the brain that affects the striatum, which is a key structure involved in motor coordination. We now know that a lot of the treatments that are available for it are based upon this initial deep insight. Huntington's disease was discovered 50 years later, about 1870. George Huntington, a Columbia-trained physician, very astute, had the insight, looking at families in Long Island, that it was a hereditary disease that he encountered, which also affected motor movement. And the defining features of that were that it was genetic, hereditary disease, number 1. That number 2, there were adventitious involuntary movements. And number 3, that it went beyond the motor system to also affect cognition and sometimes alterations in personality. The clinical picture alone suggests this disease goes beyond the striatum. It also involves the cerebral cortex. It involves the hippocampus, a structure involved in memory storage, and sometimes hypothalamus, as well as the cerebellum. Progress in this disease also was very, very slow, and it was really until about 1968 that one began to make progress in it, and this owes a great debt to the Hereditary Disease Foundation, which Milton Wexler founded. Milton Wexler was a very well-known psychoanalyst living in Los Angeles whose wife had Huntington's disease. So he started a foundation with a double purpose in mind. One is to get resources to do basic science, and the other was to try to direct the science in a productive way. And since this was a hereditary disease, the key idea was to try to define the gene that was responsible for Huntington's disease. This was in the early years of trying to do gene sequencing and gene cloning. So this is a very daring effort. But in 1983, they were able to localize the gene for Huntington's disease to chromosome 4, to the very tip of the chromosome. An international consortium of sort of gene hunters formed, and in 1993 they isolated and sequenced the gene. That was an enormous advance, because with the gene they could pop it into mice and into flies and into worms and study, mechanisms of pathogenesis, how the gene does its harm. And they learned an enormous amount about this. Moreover, as they began to look at these disorders, they realized that in addition to the clinical similarity, they both affect motor systems and they both share certain anatomical features in common. There also is an interesting similarity in the way the proteins that are involved in the disease become abnormal. They realized this is a protein folding disorder and that it belonged to a larger family of protein folding disorders, which were pioneered by Stan Prusiner. Stan Prusiner described Jakob Creutzfeldt disease many years before that as a disease that involves protein misfolding, and we now realize that in addition to Jakob Creutzfeldt disease, Alzheimer's disease that you and I talked about, frontotemporal dementia, in addition to Parkinson's disease and Huntington's disease, are protein folding disorders. What does that mean? That means the protein can exist in 2 conformations. One is the normal conformation, in which it does its normal physiological function, but it flips into the abnormal conformation. It forms aggregates, and these bundles of proteins are toxic. They kill cells. Moreover, they're also released by the cell, taken up by other cells, and in turn, these aggregates kill the cells that take them up. So this is a way of propagating the disease, extending it throughout different parts of the nervous system. This is a profound insight, which allows us to think that there may be a unifying theme connecting these disorders and perhaps allowing us to get a completely new therapeutic approach to this.